So we continue on to our next unit. Okay, thank you. And talking about cell structure and function. So we're moving up those levels of organization that we talked about in the beginning of the class. We talked about it's the most basic unit of matter is the what? Matter. Atom, yeah. So atoms come together to form molecules and compounds, right? So we covered the first two levels of basic levels of organization. We talked about the atoms when we talked about chemistry. And then we talked about different molecules, right? We talked about proteins and fats, and we talked about the importance of water and carbohydrates, right, in the body. So now we're moving up. So it goes atoms, molecules, cell. So now we're going to talk about the cell. And that's the fundamental unit of life. So it's not a trick question. The basic unit of life is the cell, because proteins aren't living, right? Carbohydrates are not living things. The cell is a combination of molecules that we're going to find. We're going to find that there's fats, which are lipids, carbohydrates, and proteins that make up the cell. So the fundamental unit of life is the cell. All right, so all organisms are made up of cells. Some organisms are only one cell. Can you think of some? What's a living thing that is only one cell big? Bacteria is a perfect example. And that's our most basic unit of life. Very simple cellular organisms are bacteria. They don't have a lot of other organelles, and we're going to talk about that. Someone said something else? Can you think of, like, maybe in biology, maybe you watched a video, or maybe you're able to look through a microscope, talking about little unicellular organisms that are swimming through a pond. Remember the names of any of those organisms? Some get into our food and make us really sick, but they're not bacteria. Any idea? Have you heard of amoebas before? An amoeba is a single-celled organism. Paramecium, have you heard of paramecium before? They have the cilia around the edge. Those are single-celled organisms. So some are just one cell big, but like us, and kingdom or class Mammalia is all multicellular organisms, right? So the cell is the simplest collection of matter that can live, so it's the basic unit of life. And when we look at the structure of cells, it gives us some idea about their function, and we'll give you examples of that. And when we look at how cells develop and how they're related, we can look at their ancestry and see how certain cells came from other cells over time. So prokaryotic cells, when we use the term prokaryote, it means these are the simplest organisms. They have no nucleus. And not having a nucleus really limits their function. So they're not very responsive to their environment because there's no nucleus. There's no, machinery to respond in a very complex way, so they're very simple. The DNA, because there's no nucleus, is not bound in a nucleus. It's just kind of a tangle of genetic information in a mass of DNA, which we call nucleoid. There's no membrane-bound organelles, but they do have cytoplasm, which is the fluid inside the cell, and they do have a plasma membrane. They have an outer covering. And we talked about that's a functional characteristic of life, right? It's to maintain boundaries. So they have to have a plasma membrane to maintain their boundary. So these are the simplest organisms. And again, what big word could you write on here that's the classic example of prokaryotic cells? You already mentioned it. What's the simplest cell that create havoc in our lives? Bacteria. So when you think of prokaryotic cells, bam, you should instantly think of bacteria. Classic example of prokaryotic cells. So we have a diagram here of a typical bacterial cell. So it has a plasma membrane. And they have little sticky extensions, little hair-like structures we call fimbriae. So think of it as little fingers, and that's how they attach to surfaces and attach to our cells, attach to our surface, our mucous membranes, our skin. That's how they can infect. So as with these fimbriae. And then this tangle of bacteria, looks like a pile of spaghetti, that's the nucleoid. That's the genetic information that allows them to reproduce and have the same species of bacteria in the next population. And they can reproduce very quickly. We'll talk about that when we get into bacteria in a little more detail. They do have an organelle. It's a ribosome, and that's where protein synthesis occurs. But it's not a membrane-bound organelle. Ribosome is a very simple organelle in which protein synthesis, like I said, occurs. They have this plasma membrane, which is the inner lining here, because the outer lining is called a capsule. And what the capsule does is it protects that bacteria in harsher environments. So when it's really dry, there's not a lot of moisture, these bacterial cells don't dry out because of that capsule that keeps them protected from harsher environments. So examples of bacteria that you've heard of probably are E. coli, right? E. coli is a classic one found in our digestive tract. It serves a healthy function, but it also causes illness if it gets in our food, right? So it has to stay in the digestive tract. We don't want E. coli to get into sterile areas like the urinary tract. It's the #1 cause of urinary tract infections is E. coli coming from the anal area into the urinary tract. That's causes UTIs or urinary tract infections, and that can lead to kidney infections, and then it can go to the blood from there. So we have people that come in from the nursing home into the hospital that have a blood-borne infection of E. coli causing inflammation of the brain where they act like they have Alzheimer's even because their brain is inflamed. We call that encephalopathy because of this infection just from a simple urinary tract infection. So it's a very common problem in the elderly. And then we just treat it with antibiotics and they're fine. Food poisoning, classic bacterial organisms in food poisoning. Common ones are E. coli. Someone didn't wash their hands after having a bowel movement and went back, and the warm environment of the food, like in fast food joints, really promotes bacterial growth. And the bacteria get their nutrition from the food in the warming trays. So salmonella, you've heard of salmonella, that's common in lunch meats. Listeria is common in lunch meats and turkey. Like I said, E. coli, and then some of the different viruses, which are not bacteria, also can cause major GI issues in close quarters. You've heard of the norovirus, common cause of the stomach. We call it the flu, but it's actually a norovirus that causes flus in close contained areas like in nursing homes, schools where half the school is out because of vomiting and diarrhea. That's the norovirus. That's not influenza. That's a completely different virus. That's a respiratory illness, influenza is. But bacteria can cause a lot of issues. But bacteria can do good things too. We have healthy bacteria in our gut that keep our gut properly functioning, and we call that our flora. Have you heard the word healthy or normal flora? So we have bacteria on our skin that protects us from infection. It's a staph bacteria. It's called Staphylococcus epidermidis, and it's on our skin, keeps us healthy. But the bad staph that's out there is Staph aureus, and that's an unhealthy disease-causing bacteria. Have you heard of MRSA? Methicillin-resistant Staph aureus. That means that Staph aureus does not respond to the antibiotic methicillin, which is a really powerful antibiotic. So it's a resistant bacteria, causes a lot of skin infections. Sometimes it infects people, it colonizes them where they don't have active illness, but they harbor the bacteria. Sometimes it's in their sputum, so it lives in their airways, it lives in their nose, it lives in urine, so old wounds, but typically when we have a wound and it has MRSA in it, we would need to be treated for that. So if you ever cut yourself, it's really important now more than ever before, like especially if you're in a crowded area or you're in the river and you cut yourself, it's really important to wash that wound out and put some antibiotic ointment on it right away. Because we see so many young college age people coming into the hospital with skin infections with MRSA, because they've acquired it out in the environment and they didn't clean it up right away, and then that infection kind of takes over. And you can end up in the hospital on IV antibiotics with a severe, illness. We call it sepsis, when bacteria enters the blood and makes you sick all over. So look at your wounds. If they're pink or turning red and warm and painful, you know what a cut is like, right? It should get better every day. It shouldn't start to get angry and red and painful. Then that's a sign that you need some more help other than just home care. Okay, so this is our simplest of cells, and the flagella, again, can help it propel itself in a moist environment. A cell wall provides extra protection, again, so it has a plasma membrane, a cell wall, and in some cases a capsule to keep it contained and surviving in harsh environments. And some of these organisms are so resistant and need so little from the environment to survive that we have found bacteria, like a really bad one in the medical environment is called Pseudomonas aeruginosa. That has been found in sterile water. I mean, it's not sterile, obviously, right? But I mean, if we take sterile water, put it in a container, that Pseudomonas can survive in that. So we really are constantly working on trying to contain some of these disease-causing bacteria. Yeah, it does look like there's a little bit of a glare. So eukaryotic cells are all other cells. Any cell that has a nucleus has membrane-bound organelles. It also has cytoplasm, just like bacteria. So they share that characteristic in common. Bacteria have a plasma membrane, so do eukaryotic cells. And because of that, eukaryotic cells are much larger than bacterial cells. So when we want to look at bacteria, what microscope lens do you think we're going to have to use when we study the bacteria coming up in a couple of weeks? Which one? What's the strongest lens we've looked at on our microscope? I wish. It's very expensive. We have just a light microscope. Yeah, but you can see a lot of detail with that. The 40 was high power. You have to go even beyond that. Do you remember what special stuff we have to use when we use that very powerful, what? Yeah, the oil immersion lens. So that's the 100x. So that is oil immersion. We have to use that to look at bacteria. And we have to stain them with a dye to see them. So we'll be doing that in lab this semester. So eukaryotic cells are much larger. So here's a transmission electron microscope showing the plasma membrane. A lot of detail there when we're focusing in on a cell and looking at the level of the plasma membrane. But it looks pretty basic in this transmission electron or TEM view. But if we look at a cartoon diagram of what the details are, there's a lot going on in that plasma membrane, a lot more than you would imagine. So the brick and mortar of the plasma membrane are these phospholipid molecules. Do you remember talking about that in the chemistry of life unit? It has the water-loving heads and the water-fearing tails. And we talked about that. I'll just, on page 31 of your notes, that phospholipid molecule. So this is the phosphate, this portion, and these are the fatty acid tails of that phospholipid molecule. So this does not allow anything that is water soluble. Anything with a positive or negative charge cannot diffuse through this oily center. But things are attracted to the surface because the phosphate head does attract water-soluble molecules. So that's good. We want things like glucose and electrolytes like salt, sodium chloride, potassium, calcium. We want those things to be attracted to the surface, but then they're going to need some kind of protein to get them across the membrane to bridge that fatty, resistant, hydrophobic core. So we have proteins embedded in the membrane. We have carbohydrates that are attached to some of those proteins, and they all have very specific structures, or functions, I mean. So looking at the functions of some of these structures, the carbohydrates that extend off the surface are for recognition. They help the cells identify each other. Like your face is unique, and once I get to know you guys in lab a little more, I can identify the name on this paper with an individual's face. So that's your recognition is the pattern of the facial features that you have, that you've inherited. A cell recognizes another cell by the pattern of carbohydrates on the surface. So the sperm finds the egg based on the pattern of carbohydrates on the surface of that egg, for fertilization. And cells, immune cells, recognize foreign invaders by the pattern of carbohydrates on the surface. There's more detail beyond that, but that's just the basic function. And then proteins can function as enzymes. They can function as transport proteins. So it just depends on the protein, how it works. So when you look at cells, and surface area is important. For example, if I was going to operate a UPS store or a FedEx store, and my job was to receive things at the doorway, people bring in their package, right? And then I put it on the shelf, and then someone else scans it in, and then I put it in the truck, and it gets shipped away to where it's going, right? Would it make sense for me to have the entrance here, and then 100 yards down the line have my shelf, and then 100 yards down the line have my shipping out area, my dock? No, that would not be efficient, right? So when cells get too big, efficiency goes down. So what happens to cells when they get to a certain size? They divide. And do you remember that process of cell division, what that's called? When cells, they grow, and then boom, they split into two. Say it louder. Mitosis. Yeah, mitosis is the process of cell division. Because what happens is when surface to volume ratio is what we're talking about. When the surface area decreases compared to the volume, efficiency of that cell goes down. So if I look at the surface to volume ratio of this cell, so surface area to volume, compared to this, look at the difference. This is 1.2. So if I have an organism made up of one huge cell versus this large cell dividing into small cells, it's the same size, right? Same overall size. But my surface area to volume is a lot larger here. Surface to volume ratio is a lot larger here, so efficiency is better. So we want more surface area than volume so we can have coming and going, because the plasma membrane is all about bringing in nutrients, giving off waste, responding and communicating with the environment, because we need nutrients in the cell, and the cell produces waste, so we have to have more surface than volume for efficiency. So that's why this box and this would be a multicellular organism, right? Made up of lots of cells. And that's why that's important. One big cell doesn't maintain efficiency. So by having things be made up of multiple cells, we have a greater surface to volume ratio. Okay, so inside of the cell then, we have this cytoskeleton. And this is what the organelles attach to. It gives shape to our cell. So it's not just cytoplasm and then these organelles floating around in there randomly. We have a skeleton that provides support, so do our individual cells, made up of tiny protein filaments. And we also have centrioles. Centrioles are structures that help with cell division. Do you remember in mitosis, the centrioles that produce the spindle, which the chromosomes attach to, and then it pulls apart? I'll show you a quick animation of that.
Mitosis.
So these are so here's a cell getting ready to divide. Here's a chameleon or lizard that's lost its tail, needs a new one, so it's going to divide. So this is the centriole, these structures here on the end. That's a centriole. This is a centriole. I'm not going to go into the stages of mitosis, but I just want to show you the growth of that spindle. So these are the centrioles that grow this spindle here. You can see it's starting to grow, and it's going to attach to these chromosomes, and it's going to push those chromosomes to the middle, and then it separates those chromosomes as it retracts and shrinks. So this is the spindle, and these are the centrioles that created that spindle to separate the chromosomes during cell division. So functions of the spindle are to separate chromosomes during mitosis. Lost my notes. Where did they go? There we go. And I better shut that down. Okay, so the centrosome is just a cluster in which the centrioles are contained, and then they separate, like I said, during mitosis and secrete that spindle. So centrosome, centrioles, same function. Cilia and flagella we need for a couple of different functions. If we're talking about unicellular organisms like the sperm or the paramecium, or we looked at the bacteria, they can use cilia or flagella for locomotion to actually move the cell. But the cells of our respiratory tract have cilia, and the function of the cilia in the respiratory tract is to move substances over the surface. So we have mucus that's produced by cells in the respiratory tract, and then the cilia beats that mucus upward to our throat, to where we occasionally feel that collecting around our larynx, and then we cough or go and then we swallow it down. So in the human body, cilia is used to move substances over the surface. So flagella for locomotion, sperm's classic example. And again, moving things over the surface, we can have cilia have a function for that, or cilia on the surface for, sometimes filter feeders have short projections that allow them to kind of interact with the aqueous environment and bring in food that way. So a variety of functions for cilia. It could be moving fluids over the surface, like in a respiratory tract, or the paramecium has cilia all around the external surface for moving around in pond water. And then we have microvilli. Microvilli they don't move, but what they are is little extensions of the plasma membrane that increase the surface area of that cell. Like your intestinal cells have microvilli, because if you have a protein that was been broken down to amino acids, and you need to absorb those amino acids, as they pass through your intestinal tract, if these were not broken up into little microvilli, all you would have is this surface area from left to right, right? You'd have just this surface area. But with the finger-like extensions of microvilli, you have all this surface area. You have the entire surface of each microvilli for absorption of those amino acids to occur. So it really increases surface area. So these are not for movement, it's just to increase the surface area for absorbing substances that are passing by these cells. Yes? It's on the outside of the cell. Good question. Yeah, it's the plasma membrane. So it's like taking your hand in a fist. You have just the top of the knuckles. That's the plasma membrane. If you extend those fingers up, that's the plasma membrane now. So you have all that surface area for absorption of the cell. So sometimes we have cell or structures that are outside the plasma membrane, so we call them extracellular components. So they're outside of our plasma membrane. So plant cells and bacteria cells have a cell wall. So that is not common to all cells. All cells have a plasma membrane. All cells have DNA. All cells have cytoplasm, but only bacteria and plant cells have a cell wall, which is that extra layer for protection. And the cell wall in plants is for structure and support, because what major polysaccharide, carbohydrate, did we say makes up the cell wall of plants that we can't digest, but we need it for roughage? Cellulose, yeah, cellulose. So that's what makes up the cell wall of plants. So fungi also have cell walls. What are their cell walls made up of? Mushrooms. They're not crunchy. Starts with a C. Do you remember the polysaccharide we studied that makes up exoskeletons of insects and crabs, and then the fungi? Chitin, yeah, close, very good, chitin. All right, so the extracellular matrix is kind of just this extra stuff on the outside of cells that again allows it to attach to other cells and keep that intact. So it's just extra little protein fibers on the outside of cells for extra support and stability. And then we have what's called intercellular junctions. Like plant cells have passageways for substances to travel between them, and we'll study that in detail when we get into lab and looking closer at cells, but these tight connecting points for cells to communicate with one another. They're called intercellular junctions. Inter meaning between cells, so they're just doorways between cells. Sometimes they're not doorways, but they're just tethering points. They're called desmosomes. You don't need to go know the detail of that, but it just protects things from entering into an area that is kind of partitioned off from the rest of the body. For example, our brain cells are tightly tethered together. They form what's called tight junctions. So when we have capillaries bringing in all the different substances from our food, maybe we have infection going on, our brain cells are very tightly tied together so nothing can leak between those cells and enter into that tissue. Our intestinal tract is not like that. Have you heard of leaky gut syndrome? Sometimes people get a breakdown in the barrier of their gut from infection, or Lyme's disease is a classic cause of leaky gut symptoms. Women sometimes have what's called irritable bowel syndrome, where they have trouble with constipation or diarrhea related to stress. They can end up with leaky gut syndrome, and then things can pass between intestinal cells and cause inflammation and digestive problems. So we have these extra things that some cells have that others don't. So again, the cell wall of plant cells are unique from animal cells. Cell walls are not present in animal cells. We have a skeleton that gives us support, an internal skeleton. It maintains its shape, prevents excess water uptake, so that's impermeable to water. And it's cellulose that those plant cell walls are made of, so make sure you remember that. This is just a picture of the cell wall. So some of these connecting junctions, plasmodesmata is what allows fluid to flow between cells. That's a special structure we'll talk about. So the extracellular matrix, like I said, are special proteins that are on the outside of cells. Some cells are receptors. You don't need to know the details of collagen, proteoglycans, fibronectin, or integrins. That's just supportive information, but you don't have to be studying those for our test. But this is what it looks like. So here's the plasma membrane here, and here's this extracellular matrix. Again, some act as receptors for other things to bind, and some just help connect this cell to the cell nearby. So they have a variety of functions. So when you look at all the organelles and where they're found, we see ribosomes are found in both bacterial cells and plant and animal cells. The cytoskeleton, found in all eukaryotes. Centrosome, that's important for cell division, but we find that only in animal cells. The nucleus is found in all eukaryotes, so all cells. We have a rough and smooth ER, so it depends. The smooth ER is in charge of making lipids. The rough ER has ribosomes attached to the outside, and that's for making proteins. And we find that in all plant and animal cells. Golgi apparatus, that's for packaging proteins and lipids, so it works with the ER, either the rough or smooth. We find that in most cells, but not all. Lysosomes help break down, they're kind of like the waste treatment plant. They break down old organelles. They contain digestive enzymes, like in the pancreas. It releases those lysosomes to break down food products in the small intestine. Peroxisomes contain hydrogen peroxide, and they help neutralize toxins in the cell. Vacuoles are large storage organelles. So there's a food vacuole and there's a water vacuole. Water vacuoles are really large in plant cells. Vesicles are any little membrane-bound organelle that contains something. So a lysosome can also be called a vesicle because it's a little membrane-bound containing a digestive enzyme. But a vesicle can contain any substance. We'll talk more about that and give you some examples. And then the mitochondria we already talked about in the beginning is the powerhouse of the cell. It makes ATP. So it takes the sugars that amino acids, and fatty acids and glycerol from the fats we eat, breaks it down, and we can make ATP with those fuels. And then chloroplasts are for photosynthesis, so of course we're going to only see those in cells that contain chlorophyll and can make food from the sun. So plants, algae, and some protozoa that are algae-like. So here's a typical plant cell. See this large vacuole here? That's what gives that plant support, and as long as we're giving our plant enough water, that vacuole is nice and plump and full. But as it shrinks, the cell wall pulls in, and what happens to our plant? It gets limp, right? And the leaves start to droop. It doesn't stand upright anymore, and then we say, oh, my plant needs water, right? And then you fill it, or give it water, and then that gets plumped up again, and then it's okay. But when it gets so bad, when you really neglect a plant, sometimes the cell membrane pulls away from the cell wall. We call that plasmolysis, and then the cells are dead, and you can't recover it by watering it. Then it's a goner. So that's the water vacuole. Here are the chloroplasts. You can see those green structures. That's unique to plant cells. And if we look at an animal cell, here's the centrioles. They kind of look like churros, or kind of like little bits of licorice, right? So, oh yeah, yeah. And they secrete the spindle, right? So these are unique to animal cells. The mitochondria, kind of classic kidney bean-shaped structures with a membrane. The rough ER always is attached to the nucleus. See how it attaches to the nucleus here? And it has these little beads on the surface. Those are ribosomes for making protein. So the rough ER is in charge of protein. Here's the smooth ER. It's the same color. It's a network of membranes. It's in charge of manufacturing lipids. It also has other functions, but that's a basic function. If we're trying to make a fatty molecule like estrogen or testosterone, this is the smooth ER's job. If we're making protein, that's the rough ER's job. And then the Golgi works with these two organelles to package those lipids or proteins and export them out of the cell. So here's a vesicle. Any membrane-bound organelle that has something inside of it, we call a vesicle. And that vesicle could be a lysosome. If it contains a digestive enzyme, then we call it a lysosome. So it's a different color here just so you can distinguish it as being something different, but it could have come from the Golgi, because if the rough ER made that digestive enzyme, the Golgi packaged it and modified it and packaged it into this little vesicle, and it contains that digestive enzyme, we call it a lysosome. Let's see. This is the large nucleus covered by a nuclear membrane that has nuclear pores in it. Inside the nucleus is another smaller round structure. Sometimes there's more than one. It's called nucleolus. And the job of the nucleolus is to make ribosomes. So the ribosomes are produced in the nucleolus. And then this hair-like stuff in here is called chromatin, which is DNA. And when a cell gets ready to divide, that chromatin will coil up into chromosomes. So chromatin, it's all DNA, but it's loosely bound. And when it coils up to form chromosomes, then the cell's ready to divide. So this is all chromatin, which is just DNA. So peroxisome is another type of vesicle, but it contains hydrogen peroxide for neutralizing toxins. Here's the microvilli we talked about. Microvilli, what is their function for the cell? Very good, to increase the surface area for absorbing things into the cell. All right, so I think the rest of the stuff we kind of talked about already. Those are maybe something less familiar. So I have a handout that has you comparing cells There's a copy of it in Blackboard, and I'd recommend that you fill this out, label the different structures on the plant, animal, and bacterial cell, and then write a brief function of each organelle, and then identify if it's found in, check off in the table if it's which cell it's found in. And that's a good preparation for our test on cell organelles.